The overall goal of this procedure is to detect GLUT4 translocation to the plasma membrane of mammalian cells in response to insulin exposure. Serum-starved myoblasts, which possess only intravesicular GLUT4 protein, are first plated in a 24-well culture dish. Antibody against an external epitope of GLUT4, pre-incubated with a fluorophore conjugated secondary antibody, is then added to the cells, followed by insulin. Engagement of the insulin receptors at the surface of the cells results in translocation of GLUT4 to the plasma membrane. Surface GLUT4 is then recognized by antibodies in the medium. At this point, the cells can be fixed and the intensity of fluorescent staining measured by flow cytometry.
This method can be used to study GLUT4 translocation and glucose metabolism, but also can be applied to other systems such as NK cell degranulation through exposure of CD107A at the membrane.
For this procedure, it is important to use cells that are actively growing or 60 to 80% confluent and have been serum starved overnight. After harvesting the cells by trypsinization, wash and resuspend them in regular growth medium at 200,000 cells per milliliter. Plate the cells in a 24-well plate at 0.1 million per 500 microliters per well. Then, incubate the cells for 1 hour at 37 degrees Celsius to allow cells to recover from trypsinization. In the meantime, mix the primary anti-GLUT4 antibody and the Elexaflur 488 conjugated secondary chicken anti-goat IgG antibody in a 5:1 ratio. Calculate the final volume needed, taking into account that 10 microliters of the antibody mix will be used for each well. Incubate the antibody mix for 10 minutes at room temperature in the dark. While the antibody mixture is incubating, prepare a 2 times working stock of insulin by diluting it to the desired concentration in growth medium. Once the cells have incubated for 1 hour, remove them from the incubator. Gently add 10 microliters of antibody solution to each well containing cells. Be sure to leave at least 1 well unstained. This negative sample will be used later to set up the flow cytometer. Next, add 500 microliters of the 2 times insulin stock to each well to trigger translocation of GLUT4 to the cell surface, and incubate the plate for 30 minutes at 37 degrees Celsius in the dark. After removing cells from the incubator, fix them by adding 1 milliliter of PBS 1% PFA to each well, and incubate the plate for 20 minutes at room temperature in the dark. Using a cell lifter, gently detach the fixed cells from the bottom of the wells and transfer them from each well to an individual fax tube. Centrifuge the tubes to pellet the cells. Then, vortex and resuspend them in 1 milliliter of PBS to wash and centrifuge the cells again. Repeat the wash step twice, discarding the supernatant between washes. Finally, resuspend the washed cells in 400 microliters of PBS 1% PFA. The cells can now be analyzed for surface expression of GLUT4 in the flow cytometer. Once you've arrived at the flow cytometer with your samples, begin acquiring data from the unstained control. Define a broad live cell gate using the forward and side scatter parameters. Then, adjust the voltage setting for the FL1 channel so that the negative peak is visible within the margins of the histogram. Once these parameters have been set, run each experimental sample, recording at least 10,000 live events per tube. This histogram shows the upregulation of surface GLUT4 staining on myoblasts exposed to 0, 1, 10 or 100 nanomolar insulin. In contrast, myoblasts from insulin-resistant individuals fail to upregulate GLUT4 in response to 100 nanomolar insulin. If the mean fluorescent intensities of stimulated cells is normalized to that of unstimulated cells, it is possible to compare the healthy and insulin-resistant myoblast MFIs directly.
Using this procedure, cells can be stained for additional surface or intracellular markers to identify different cell subsets in mixed populations.